Hello dear students, welcome back to plus 2 biology. So biotechnology applications in agriculture, and in the topic we have discussed the Bt gene transfer and production of transgenic plants, Bt plants, and our aim was to produce insect resistant plants. So by transferring Bt genes to various crops, we could produce number of insect resistant GM crops, Bt plants like Bt rice, Bt brinjal, Bt cotton, Bt soybean, okay. And our next concept, how to introduce pest resistance in plants by RNA interference. Development of pest resistance in plants by RNA interference technique. So RNAi is expanded as RNA interference. So what is RNA interference? Interfering RNA. And this is not a fully artificial method. This is not a new technique. See, majority of the eukaryotes already adopt this technique as a method of cellular defense. This is a naturally occurring phenomenon in many eukaryotes. What do you mean by that? Interfering RNA. What do you mean by that? So, whenever any pathogen attack our body or any other organism's body, it enters the body of the host, means its DNA will be expressed to RNA, then will be expressed to toxic protein. That means a particular segment, which is the DNA segment, okay, forming the protein, will be first transcribed to RNA, then it is translated to the toxic protein. You know the fact. But here what happens? In cellular defense mechanism, in RNA interference technique, when the pathogen enters the host is having a defense mechanism in which it may allow the pathogen's DNA to form RNA, but the RNA is not translated to protein because the RNA is blocked or interfered. We can call one more term is there, RNA is silenced. So this is also known as RNA silencing. How can the RNA be silenced? How can the RNA be silenced? Tell me. Normally you have studied a double-stranded DNA, okay? Then it is first transcribed to an RNA, okay? One of the two strands get copied to an RNA. Suppose this particular segment is the toxic gene, the gene producing the toxin. Then it produces the corresponding RNA, and when this is translated, it produces the toxin. This is what is natural. But what is happening here is, here the RNA is blocked. How can the single-stranded RNA be blocked? In order to block that RNA, a complementary RNA copy is required. So the RNA here, the pathogenic RNA is blocked due to the presence of another complementary RNA. So that it forms a double-stranded RNA. A double-stranded RNA can never take part in translation. It can never produce the toxic protein. Okay? Keep the fact in mind. Here, this is a gene which produces toxin, a pathogenic gene which produces toxin. Normally, it is transcribed to RNA, then it is translated to the toxin. Okay? Whereas in RNA interference, when this strategy is there in the eukaryotic organism, a particular organism with this strategy will be having a complementary RNA, and this is blocking this pathogenic RNA. And the source for this complementary RNA, okay, where this comes from where this complementary RNA comes from. It may be, the source may be either through a viral infection, okay, it may be there in the plant or animal, the organism. It may be through a viral infection, or it may be produced, it may be available with the help of transposons, mobile genetic elements. Okay, so the source of the double-stranded RNA may be either a viral RNA or transposons, mobile genetic elements. Okay. So this is there naturally in some eukaryotes. I mean the RNA is silenced and is made double-stranded. It is not allowed to express to form the toxin. And this strategy is adopted to develop pest resistance in plants. Okay? I mean, in order to develop pest resistance in plants, suppose this is a pest gene and is producing the toxic RNA and this is producing toxin. This is attacking the plant. Okay, causing damage to the plant. And in order to avoid the production of the toxic protein to plant, we can silence the RNA. So this technique is adopted in what? rDNA technology. How is it carried out? Okay. So, okay, if these things are clear, we can just proceed to how this technique is used by rDNA technology. This is a technique naturally in plants and animals and eukaryotes. Okay, how this is taken into rDNA technology. Okay, the typical example is development of pest-resistant tobacco. Okay, nematode-resistant tobacco. A typical example for RNAi developed by rDNA technology is development of pest-resistant tobacco. Okay, and a particular pest is a nematode worm, Meloidigine incognitia. Meloidigine incognitia. Okay, so listen here. In order to protect tobacco plants from this particular nematode worm, nematode. Okay. In order to protect tobacco plants from Meloidigine incognitia, which technique is used? RNA interference technique is used. So for that, what is done in this case? At first, the plant is introduced with the help of, in order to transfer to plant, what is required? Agrobacterium vector. Its plasmid is required. Okay, using agrobacterium, the plant is delivered with the particular gene. I mean the pathogenic gene of this Meloidigine incognitia worm. Okay, through agrobacterium, nematode specific gene is introduced to plants. Listen, in the first step, with the help of agrobacterium as vector, nematode specific gene is introduced to tobacco plants. Okay, that is the first step. Then, in this particular introduction, we are providing conditions so that this nematode gene will produce both sense and antisense RNA. We should provide such conditions. Okay. So, by the time of its introduction, it is made sure that this gene will produce. Normally, DNA will produce only one strand of RNA. Here it is made that it will produce 2 strands of RNA. Okay. So, it produces 2 strands of RNA. If 2 strands of RNA are produced from 1 strand of DNA, we know that they are complementary, so it will form a double-stranded RNA. Okay, so the plant is now possessing double-stranded RNA inside. Just imagine when we take vaccine in order to get protection from pathogens, we are taking the pathogen as such in the attenuated form or its toxin. Similarly, in order to get protection from the nematode specific gene is introduced to plant. Likewise, we use vaccine. Okay, the nematode gene is introduced to the plant which we need the protection. Okay, and with the help of a vector agrobacterium, but we should have some formulations. Likewise, we attenuate the vaccine. Vaccine is not actually the pathogen as such. We are doing some treatment. Similarly, what this treatment is? We are giving conditions for the gene to express and produce. It's both the RNAs, both the templates are copied to RNA. So, if sense and antisense strands are formed, and it forms immediately a double-stranded RNA. Okay. Then now the plant is with the double-stranded RNA. When we take vaccine, our blood will be with the antibodies, produced antibodies. Similarly, here what will be there? Okay, when we take vaccine, our immune system is producing antibodies against it and keeping it as memory. Okay, to fight it for the next time attack. Similarly, here instead of antibodies, what is produced in the plant is, it is producing what? Double-stranded RNA, and this double-stranded RNA will be kept in the plant. Okay, so the plant is now transgenic. Okay, it is a processed plant. Then we are moving to the next case. Suppose such type of a tobacco plant. Okay, just a small diagram. Okay, this is a leaf. Okay, just think. Suppose this is a tobacco plant. Okay, and this is its root system. And this nematode, Meladygene incognitia, it's a root parasite. Okay, it's the one which inhabits the soil and it enters the root through the soil. And see, we have treated this plant with this is a transgenic tobacco now, and so in this plant there is what? Double-stranded RNA. Through agrobacterium we have introduced the nematode gene, and now it is possessing what? Possessing what? Double-stranded RNA inside. It's having the double-stranded RNA inside. Okay. Likewise, if we have taken chickenpox vaccine against chickenpox, the antibodies against chickenpox will be there in our blood. Likewise, it is there in the plant. Then when we get the chickenpox, when a chickenpox virus attack our body, what happened? Our antibodies go and fight against it. Okay. Similarly, when the root parasite, when the worm enters, what happened? This worm, when this nematode worm enters the plant through the root. Okay. Suppose the worm entered here. Okay. How it is not allowed to act? See. Then suppose a worm enters here and its DNA is forming its RNA. Okay. The nematode worm, okay, when it enters the plant, transgenic plant now, its DNA is there and it is forming its RNA. Normally single-stranded RNA only it forms. Okay. But immediately it is forming, its DNA is copied to its RNA. But immediately what happens to this double-stranded RNA already existing here? Okay, this double-stranded RNA, these are complementary to the nematode RNA because it's highly specific, produced by introducing nematode gene. So, this double-stranded RNA will be immediately cleaved into fragments. Okay. So this function as a unit and this will be cleaved into fragments. Okay. And these fragments will work as a complex and this is known as RISC. RNA induced silencing complexes. RNA induced silencing complex. We will discuss it. Here is in order to silence the RNA, a complex working. They are working as a complex. See, the double-stranded RNA then will be cleaved into fragments and they work as RISC. RNA-induced silencing complex. What it consists of? It consists of small RNAs. Okay. This consists of number of small RNAs. Fragments of RNAs. We can call it as small interfering RNA. Okay, these are named as small interfering RNA. Okay, small interfering RNA. Then what happens? Tell me what happens. So, this small interfering RNA will go and bind to the nematode RNA. It is so this siRNA go and block the nematode RNA, and this is blocked by this is known as silencing or RNAi. This technique is known as RNA interference, RNAi. Okay, so this is a technique to develop pest resistance in plants. Okay, so once again we will discuss it and then I will just give a flowchart representation of this. Okay, it was asked to produce as a flowchart for board exam once. So listen carefully here. RNA interference or RNAi, also called RNA silencing, is a method of cellular defense seen in many eukaryotes. A natural method of cellular defense seen in many eukaryotes. In this method, the RNA of the pathogen is blocked with the help of a complementary RNA. The source of that complementary RNA may be either from the infection of a virus or through the jumping genes, mobile genes, transposons. Okay, whatever. In a eukaryote, this is seen normally. A particular RNA of pathogen is not allowed to work because it's blocked by a complementary RNA. Okay, and this technique is adopted in RNA technology to develop pest resistance in tobacco plants. Okay, in order to develop nematode resistant tobacco plants, this method is used. The particular nematode is Melodigine incognitia, which is a root parasite. Okay, then at first what is done is, with the help of the common vector used in plants, that is agrobacterium, with the help of agrobacterium, the nematode gene is introduced to a normal plant. A tobacco plant is introduced with a nematode gene through an agrobacterium vector. And we are providing K condition, that means its DNA, both the strands are acting as template and it produces sense strand of RNA and antisense of strand of RNA. So that what happened, it forms two RNAs and this being complementary forms binding to form what? A dsRNA. Double stranded RNA will be produced. Okay, the plant is now possessing, the transgenic plant is possessing double stranded RNA inside. Okay, then this plant is safe for the further attack of the worms. When a new worm attacks its root or enter through its root, when it is expressing, when its DNA is forming its RNA. Okay, this RNA is silenced, this RNA is blocked with the help of the existing double-stranded RNA. How does this antibody act here? Our antibody here is the double-stranded RNA. How does this double-stranded RNA act? Okay, this double-stranded RNA will function as a unit. Okay, it is called RNA-induced silencing complex RISC. Okay, what's the name? RISC, RNA-induced silencing complex. Then in this RISC, RNA-induced silencing complexes, the RNA is actually, double-stranded RNA is cleaved into number of small RNA fragments. This is known as siRNA. These are small interfering RNA, expanded as small interfering RNA. These siRNAs will go and bind to nematode RNA, so that the nematode RNA is silenced. Then, so this way how a vaccine gives safety to our body, from the pathogens by producing antibodies. Here, the RNA interference is giving safety by producing double-stranded RNAs. Okay, it's a simple technique and it is a novel strategy seen in naturally in eukaryotes. Okay, so clear how to develop pest resistance in plants by RNAi technique. Okay, thank you.